Hi, I'm Richard Sever from Cold Spring Harbor Laboratory and BioArchive here at the symposium, the virtual symposium from Cold Spring Harbor on biological timekeeping. And I have Ron Evans from the Salk Institute. Ron, welcome virtually to Cold Spring Harbor.
Thank you very much. Pleasure.
Well, it's nice to see you again. We were just saying I think this is the second time that I've interviewed you. So, so we will sort of move on from there. But I really enjoyed the talk that you gave during the symposium. What was kind of interesting to me was you started with this intriguing cartoon of the two strands of DNA that extended out to become a texture. And you talked about the importance of nuclear receptors in control of physiology. But from a high level, what are we talking about when we say nuclear receptors? What do we mean by that?
Well, nuclear receptors are essentially ligand-dependent transcription factors, and they're best known for the very earliest hormones that were identified 100 years ago. Were things like vitamin A, vitamin D, thyroid hormone, and steroids like corticosterone. Anyway, those were the initial hormones. No one knew how they worked. They all won Nobel Prizes for their discovery. And that opened up an era of trying to understand the chemistry of the ligands that control these physiologic pathways. What is vitamin D, or what is vitamin A, and how does that relate to steroids? And then maybe 75 years later, we had the hormones and the compounds, but we didn't understand how they work. And so it's one thing to say this is a hormone because it has this effect, and you can measure something in the blood, but that doesn't tell you how it works. It's a little bit like a computer. You can do a lot on your computer and complicated things with your screen. You don't know how it works. That doesn't tell you anything how it works. And so, and that's what we're interested in, not what you can see, because that's kind of what medicine is. Medicine works with what you can see and what you can measure. We're looking sort of behind the scenes, and the receptors are basically ligand-dependent transcription factors that modify gene activity. And so what it boils down to is what we discovered is beyond the known hormones, which are about 7 that were major ones at the time. When we discovered the first of the nuclear hormone receptors, we realized there was homology between the first one and other ones that seemed to be present in our screening technique. And in the end, what we thought would be 7 receptors opened up into a much bigger category. They became a superfamily. And so, I proposed the existence of a superfamily 3 years after we cloned the first receptor, which was the human glucocorticoid receptor. And the important thing about the superfamily is that it gave us 39 new physiologic pathways that were not even known to exist. Right. And so that leads to the nature of, what were these other ones? And that's a lot of the work that I've done. And a lot of that work ultimately impacts on 2 things. One is metabolism, and the other is circadian rhythm, which actually I believe are linked to each other. And going back to the slide that you mentioned, I think was called balancing the fat equation. And fat as its energy source is important, but also it is a potential problem in our society because obesity, we know, is a risk factor for almost every problem, whether it's diabetes or heart disease or liver disease. It's a risk for virtually every cancer. Obesity is a growing problem in the United States and in the world, and it is driving the largest part of our medical budget. And so many people are trying to build drugs to deal with it, but obesity and metabolic pathways are in large part controlled by nuclear receptors. And so the illustration is a double helix that is transitioning into a tape measure that is sort of your waist size. And inside that tape measure were lipids. And it basically said that the underpinning of all these physiological pathways are epigenetic changes in response to our environment and our diet. And so we want to dig into that relationship. How does what we eat, how do the foods we eat and the external things we do like exercise. So foods bring energy in, exercise consumes energy. And when you think about how that works, it works in a circadian fashion. When you're asleep, you're not bringing food in. Basically, you're burning energy during your sleep, but when you wake up, you eat. That brings energy in. That energy is distributed to mostly storage in your adipose depot, and that gets burned again during the day when you're active. So daylight is physical activity, and night is inactivity, sedentary behavior. And so a lot of my work is looking at activity, and part of the presentation I gave at the meeting relates to the master regulator of energy metabolism in the body, which is called PPAR delta. It is a highly oxidative nuclear receptor, which means it promotes oxidative metabolism of lipids typically, or sugar in muscle. So when you exercise, basically you burn two things. You burn sugar and fat. And the master regulator of that process is PPAR delta. It's one of the nuclear receptors that we discovered in the early 90s. I've been working on it ever since then. And as a master regulator, we've learned that it's easy to make drugs to it. We have drugs that are in clinical trials. And so I find this particular receptor, it's the heart of activation when you move.
So PPR delta, I mean, I guess for one thing, if I remember rightly, you can tell me what PPAR stands for, and that's not really going to help the conversation. Is that correct?
Right. I can tell you that. It was again, there were these orphan receptors. We had so the inversion that happened through our science is that 100 years ago, you could isolate the factors. But with the discovery receptors, you could get the receptors now, and that led to finding the factors. And so it used to be you have to have the hormones first and then find the receptor. Now we have receptors and you have to find the hormone. And so, and that created a huge activity in discovery mechanisms basically throughout the world. People got very much involved in all this and opened up a whole new era in sort of physiology, and what you might call molecular physiology. And so PPAR, it stands for peroxisome proliferator activated receptor. And it was first described by Steve Green, who said that there's three PPARs, alpha, beta, I should say alpha, gamma, and delta. That's what they're called. And they bind small molecules. One of the molecules they bound was known to induce peroxisome proliferation. Peroxisomes are entities in the cell that help to burn energy and other components in the body. And so peroxisome proliferation was just a way of saying that when you activate this particular receptor, which was now known as PPAR alpha, it triggers this response. And he was looking at it in the liver. And so that launched what became three-member family. It's called alpha, gamma, and delta. And so alpha is very active in the liver and in the heart. PPAR gamma is the master transcription factor for adipose. If you have it, you get adipose tissue. If you don't have it, no adipose. If you put it into any cell, it becomes an adipocyte. And so this is the power of nuclear receptors, is to determine functional properties in a ligand-dependent fashion. And this is key because ligands come and go with the daylight cycle. And so there's a rhythm that gets set, and that rhythm is acting on the receptors to concurrently cycle on and off and give rise to epigenetic changes in genes that lead to the activation of gene networks that really represent the molecular program for physiology. So medicine measures what's in the blood. We're looking at the physiologic response as a genomic network, not just in one cell, but a body-wide network. So the hormones go everywhere. And so you're seeing body-wide coordination. And that's why I think the receptors are very important in circadian rhythm, because how do you control body-wide functions in a smooth rhythmic fashion? And it's the easiest thing for receptors to do. Ligands come and go with the daylight cycle. Some of the ligands are actually just the foods you eat. You know, the most common energy that you get is linoleic acid. It's most of your calories on a daily basis. And that's the perfect ligand for PPAR gamma. And it's also a great ligand for PPAR delta, which was the source of my talk. And so I think that physiology is a process that occurs in space and time, but at a genomic level. And so that was kind of the background of why what we do in terms of understanding transcriptional mechanisms is the underpinning of understanding circadian rhythm. And so the one of the focuses of my research is on skeletal muscle, because that's where most energy is consumed. And the skeletal muscle, as you know, when you, for example, walk or run, becomes active, and you activate mitochondria to burn fat that creates ATP so that you can keep moving. And humans and most animals are exceptionally good at moving. That's one of the defining things. But when you're sedentary, you're going against what is a natural human property. And by being sedentary, you store that energy and you get out of rhythm. And people who are obese run into all sorts of body-wide problems because they're sort of decoupling the normal homeostatic mechanism. And so the talk was looking at a new homeostatic mechanism in the context of this larger picture. And so, which is, how do receptors cycle the activity of the genome? It then leads to body-wide physiologic effects. And typically, as I mentioned, you think it's the ligand that creates the cycle. But in this case, it turns out that there was a surprise. And the surprise was that all transcription factors attract cofactors. And some of these cofactors are repressors. Some of these cofactors are activators. And some are enzymes that modify the transcription complex. And transcription complexes have roughly 150 to 250 different proteins that form a big regulatory element. So that takes a lot of proteins to control gene activity.
You showed a very nice slide where you had the inhibition and the activation and sort of wildly different complexes in both cases. Is that correct?
That's correct. And so while PPAR delta is an activator of metabolism, and that's one of the things that we showed, and it does this in a rhythmic fashion, we thought it was going to be the ligand production that was controlling it. But it turns out PPAR delta itself does not cycle. It stays at exactly the same level but its target genes are going up and down. and we're trying, how does that work? Because in many cells, they're receptor cycles. And as that happens, it turns genes on and off because a lot of hormones induce the genes. So retinoids induce the retinoic acid receptor, it goes up and down. But PPAR delta is like rock solid, no change in the muscle. And it turns out a specific transcription factor that linked is the known regulator of non-Hodgkin's lymphoma. It's called BCL6, which it's B cell leukemia 6. And that is a well-known regulator, but it was in a cancer setting. My lab rediscovered it, not in cancer, but in associated with nuclear receptors, work by Grant Barish, who's now at Northwestern, and in macrophages. Now, macrophages are everywhere. So we thought this was a B cell specific transcription factor. It turns out it's everywhere in your body because macrophages are. And when you eliminate that factor, you get chronic and super active inflammation. Which leads me to be also interested in the link between nutrients, inflammation, and homeostatic mechanisms. And so there's a lot going on in understanding BCL6. The surprise for us was that we found it again in postdoc in the lab in muscle. I didn't expect it to be in muscle. We said it's in macrophages and in B cells. And then, in one of the lab meetings, I go, well, I discovered something new. This thing that we've already been working on is in the muscle. And that immediately attracted my interest. A good portion of my lab is on working on the control of energetics in muscle. And so when we focused on it, it turns out it's the critical regulator of PPAR delta activity, and it's a molecular break. And so it cycles, and it has a circadian rhythm. And as it cycles, it either puts the brake on PPAR delta or pulls off and lets the ligand be the accelerator. So you have a combination of almost always of an accelerator and a brake. They're alternating with you, just like you're driving a car. You're going to be alternating between, you don't do them both at the same time. In that context, the cycling rhythm of the muscle is determined by the cycling of BCL6. And so that was unexpected. And it answers a very important question, which is how does muscle become circadian entrained and link with the rest of the body? And it's not like we thought. And so it revealed a new mechanism of circadian entrainment that opens up a new way to kind of manipulate the process. And we can actually control BCL6. There's small molecules that are based on some of the work that our lab's done, and not knowing about this, we're looking at BCL6, made small molecules that could change its activity. And Ari Melnick has done a lot of work on this. And he's trying to get these drugs to go into the clinic. Initially for B-cell lymphoma, which is what I wanted to also go in the clinic for. But now we know it may have a much wider effect broadly on metabolism and metabolic disease. And in the oncogenic form in B cells, the BCL6 is the sort of the on-off switch for cell proliferation. It's a brake on cell proliferation. When it's gone or mutated, cells just keep proliferating, and that leads to leukemia. And so we knew how important this gene is in that one cell. Initially, we thought it was only in one cell, but now it's actually much more widely expressed than we thought. And so in muscle, which is the key tissue for controlling cycling rhythms, this appears to be the heart of that cycle. And so that's why I'm we're excited by it because previously we proposed that circadian rhythm is really a metabolic rhythm, and daylight rhythm or daylight energetics are just the opposite of sleeping energetics. And so the nature of controlling complexity now comes down, of this rhythm comes down to a cofactor, which is BCL6, and sort of the assembly of a nuclear receptor complex that actually determines physiologic outcome.
Well, I mean,
so,
it's a fascinating story. I mean, at one point you said that the nuclear receptors, I think you referred to them as physiology's architects. It almost seems to me that PPAR delta was sort of physiology's personal trainer in some respects. I mean, you've done some fascinating work. I think this circuit controls the type of muscle, and you know, you could make mice that never stop running. Is that correct?
Yes, and so a feature of PPAR delta that we didn't understand early on, we did know it was activated by dietary lipids, and that was important on its own because usually the ligands tell you what it does, and if it's activated by a lipid, it's controlling lipid metabolism. If it's the bile acid receptor and it's activated by a bile acid, it's controlling bile acid metabolism. And so usually the ligand tells you exactly what the physiologic response is, which is why I liked the hunt for new receptors and new ligands, because we learned a lot in a very short time. And so that put together many stories. But in this context, yes, there are a number of concurrent events in muscle that really, I think, are the hallmark of, you know, what I called in the beginning balancing the energy or the fat equation. Comes down to some of these, in some ways, simple principles. There's a lot to learn about how that works and where it can take us. But the drug development is also very active in this space. And so we have now made potent PPAR delta ligands that are apparently safe. We can give them at any level we want in animals, and we get really no adverse effects. But what we learned, as you mentioned, is if you give sedentary mice the ligand, but no exercise, and then you put them on a treadmill a month later, and they're getting a daily dose of the drug, and you run them against normal mice, also who have not been trained, but are, so they're basically sedentary mice that have, both of these mice, the first time they see the treadmill, the control mice run for about an hour and a half. And initially the mice that got the PPAR delta drug ran twice that much, 100% increase, which is a big change in physiologic response. And so instead of 1.5 hours, they're running 3 hours, which is a long time for a mouse to run. And we thought, this is the peak. This is what I call synthetic physiology. But then the postdoc was working on it, up the dose. And it turns out, instead of running 3 hours, they ran 5 hours. And then he upped the dose. Instead of running 5 hours, it ran 7 hours. And then he upped the dose. And I remember he went to the animal room to work on this particular mouse at 3 o'clock in the afternoon. And at 7, because you have to stay and watch each mouse run, you have to be there the whole time. And so it's at 7 in the evening, he calls his wife and says, honey, go ahead and have dinner with the kids. The mouse is still running. And then at 9 o'clock at night, he says, honey, put the kids to bed. I'm going to be late. And then at midnight, he says, I'm coming home. So this mouse ran 9 hours, which I think is the world record for an untrained mouse to run. And that shows you the power of these pathways. And the pathways are overbuilt so that you have the capacity. You never know what the capacity is until you really push it to the extreme. You have a capacity. It's like if you have a great music system, you may play it at a normal level, but it has the power to be played at a much higher level. You get the best quality when you have a lot of power in these systems, but you play it at a normal level. It really changes the difference than if you have a little thing with, you can hear, but the sound quality is terrible. And so when you overbuild systems, it gives you a lot of safety measures and adaptability. And that is what we learned in part from this whole process is it's not overbuilt in a random way. This is evolution, saying the more you build in redundant systems and create this robustness and resilience, the better you are. And the more random encounters, because you don't know what you're going to encounter in life. It's so many different things and you can't plan it. So you overbuild. And by doing that, you create something that's very special, which is very robust and dynamic physiology. And as I mentioned in the beginning, humans are designed to move. We are one of the best runners on the planet. There are very few endurance animals that can outperform us. I'm not counting birds because they can fly from Cape Cod to Venezuela without stopping, without drinking, without eating. That's pretty good endurance. And they get 1 million beats of their wing as they keep going. And I'm interested in that. If it's the same receptor that's allowing them the birds to get there too. So I love the nature that you can figure out and make links between what you eat as your diet, how that's managed and processed, and what the nature of physiologic mechanisms are at the fundamental level, which is in some ways a straightforward way of controlling epigenetic networks. Mm-hmm. But it's done in a way that allowed many parallel pathways to evolve. In humans, 48. So we began literally in 1984 thinking there are 7 physiologic pathways. And by 85, we knew there's many more. And by 90, we knew that there were 48. But one of the big advances is the link between these receptors and circadian rhythm. Some of the other talks touched on reverb alpha and reverb beta. That's Mitch Lazar. That's a core regulator of the circadian clock itself. So there's a clock mechanism that is dialed up and down by reverb, which is a cycling repressor. And he probably didn't talk about it, but there's a counter cycling nuclear receptor called ROR. And they go opposite. They bind to the same sequence, repressor, break, repressor, break. And they're just designed to go back and forth with each other. And so the system is basically, you reuse the same idea, you repurpose it in different ways, and you get this interesting mechanism. I do think that nuclear receptors are physiology's architect. That is, how do physiologic systems arise? I'm very interested in that question. And what's the role of nuclear receptors more generally, not just in animal physiology, but in who we are as a kingdom. So these receptors are present only in animals. It's the only transcription factor family that's only in animals. All the other transcription factor motifs are in bacteria, yeast, mold, or plants. Mm-hmm. Because animals are the last living things to evolve. And so they got the gift of everything else. But in fact, it appears that to be different, to form a new kingdom, you need something different. And I think physiology and our ability to move in our environment was the big difference. And that's where nuclear receptors come in. So they're not only physiology's the architect, basically, but I think they lie at the origin of the animal kingdom. And the nature of what we do and how we behave has to come from somewhere, and something that's new. And because these are sensors of environment and the foods we eat, that by having these sensors, you can adapt to your environment in new ways, and seek out where the nutrients are, sense them, and have this dynamic regulation by your environment that lets things turn off or on. And so I think that, from the simple ideas of how these on-off switches work, there is the bigger question of where did they come from in the beginning? And why are they so intrinsic to animals? And what's their role in our evolution and ultimately in our behavior? And so there's a ton to do, but I think circadian rhythm is at the heart. But I also think nuclear receptors are principal drivers in sort of animal behavior. And they provide a way to break it down in a unique fashion.
Well, I mean, a lot of us have been sitting on our couches for the past year and not moving very much. So I think everybody is going to be waiting for all this to translate into to human physiology. It's a fascinating story. Thanks so much for taking time to talk with us today, Ron. Thanks very much.
Thank you. Thank you very much. Appreciate it.